All right, guys, today we're going to talk about genetics. All right, now genetics right here, this is going to be the study of genes. All right, and when we talk about heredity is the study of passing on your genes. So if we think about genes, remember you get them from your mom and you get your genes from your dad. Remember we talked about how meiosis will divide up your mom and dad's genes into egg and sperm, and then they'll come together through fertilization to make that zygote. All right, so since you get them from your mom and dad, that means you get to have 2 copies of every gene. You get 2 of them. And remember, 2, the word root, that's di, so this is diploid. This is where we get diploid from, is because you get 2 copies of every gene. Now, when we talk about genes, let's go through vocab. Say gene, this is just a segment of DNA that codes for some particular trait, right? And whenever we look at our chromosomes, if we draw like a thicker X, because remember that's our chromosome, but if we draw it like this, when we start looking at chromosomes, you can see it's almost like a barcode on your chromosomes. Well, every time you see a segment, like that's a segment, and that's a segment, and that's a segment, those are literal genes. Like you can actually see genes on your chromosomes. And then those genes code for some particular trait, right? And a trait, by definition, is just a physical characteristic. It's something that you can literally see. Like, you can have freckles. That's a trait. Or you can have blue eyes. That's a trait. And so one example of a physical characteristic would be height, okay? But height is kind of a big overarching word, right? Like you don't just have one kind of height, but you can have people who are tall, you can have people who are short, and everything in between. So since you can have different versions of that trait, right? Like tall versus short, that's called an allele. Right? So an allele, by definition, is an alternative form of a gene. But really all that means is that when you look at a physical characteristic, that you can have different versions of it, right? So we call those alleles. So you can have an allele for tall, you can have an allele for short, you can have an allele to have freckles, you can have an allele to not have freckles. But since we're doing genetics, we get to use letters to denote the alleles themselves, right? I get to use letters. Well, since we're using the English language, right, I can have 2 options to write a letter. I can either be a capital letter, and I'll just use the letter A for simplicity, or I can be a lowercase letter, right? Because they're both the letter A, but these are different forms of the letter A capital and a lowercase. Now for later, when we get to it, we're going to use the capital and a lowercase to represent dominance. And so if I have a capital A, that means that's going to be the dominant trait. And what I mean by dominant, I don't mean the one that you see the most often in a population. We can talk about that later. But if I'm a dominant trait, that means I only need one to show the trait, which we'll talk about what that means. But a capital letter, A, that's an allele, and this allele shows the dominant trait, and I only need one to show it. Versus my lowercase letter, still an allele, still an alternative form of a trait, but since it's lowercase, we're going to use that to mean recessive, and since it's recessive, I need 2 to show that trait. Okay? So recall, when we were way up here, we said, hey, you get your genes from your mom and dad. So that means I have to have 2 letters, right? Because I get 2 copies of every gene. So that means I have to have 2 of those alleles, right? That's what a genotype is. A genotype is a combination of 2 alleles. And like I said, we're just going to use the letter A right now. You can use whatever letter you want. But for a genotype, for example, I can put 2 big letters together. So maybe you got 1 big letter from your mom and you got your 1 big letter from your dad. Or you could get a big letter and you can get a little letter from each of your parents. or you can get 2 little letters. Now we get to name these suckers. If we look at these 2 big letters right here, big A, right? Since they're the same, we can call those homozygous, because remember homo means same. So these 2 capital letters are homozygous because they are the same. They are both capitals. And since I have at least 1 big letter, that's going to show the dominant trait, right? Because way up here, we said, hey, you only need 1 to show it. Now this one right here, I have a big A and a little a. Well, they're not the same, so I can't call them homo, but I can use the word hetero, because hetero means different. Okay, so these are heterozygous genotype. But because it has this 1 big letter right here, it will also show the dominant trait, because again, dominance, I only need 1 to show the trait. And that's how traits can get, I won't say lost, but they can be hidden in families. Like maybe your grandma has a trait, but your parents don't, but then you do, right? That's because maybe they were carrying that recessive trait, but they showed the dominant trait. And then my last genotype are 2 little letters. Well, look, they're both little, so they're the same. So that's why I get to call them homozygous. But since they're 2 little letters, they're going to show the recessive trait, which is what we set up here. I need 2 to show the trait. All right, so I have my genotypes, but guess what? I can't see your genes, right? Like if I look at you right now, I cannot see the DNA, but I can see your physical expression of your genes. That's called your phenotype. Okay, so phenotype is the physical expression of a genotype. And so if we do a really easy example, like let's say here's my trait. Here's my trait, and my trait was height, right? Remember height, that is kind of an overarching thing. It's kind of an overarching thing. So I can have different versions of my height. I can have dominant, we'll call tall, can be the dominant trait. And then recessive can be the short, the little letter. So if I look at my genotypes, big A, I know that that's homozygous dominant, and then my phenotype that would be expressed would be tall. And if I look at my heterozygous, okay, well I have at least 1 big letter, so there's heterozygous. I know he's going to show the dominant trait, so he will also be tall. And then these 2 little letters, okay, I know he's homozygous, but since they're 2 little, it'll show the recessive, and so my phenotype would be short. All right, so now, once we have all the vocab, I actually get to use a tool called a Punnett square. Okay, a Punnett square is a device used to predict like offspring genetics, like what the offspring genotypes are going to be. And for what it's worth, there's this guy called Gregor Mendel. Gregor Mendel was an Austrian monk. He was super intelligent. He had a big laundry list of things that he did, but he chose the monastery lifestyle because he could study science, and he studied pea plants amongst other things, pea plants being the most used and recognized. But he used pea plants, and he started figuring out all of this genetic stuff, which is really stinking awesome. So a Punnett square, it looks just like that. It looks like a window pane. It's made up of 4 boxes. 1, 2, 3, 4. Okay? And so then, if I want to predict offspring, that means I need to know my mom's genotype. I need to know my dad's genotype. So for example, let's make my mom heterozygous. So she has a big A and a little a, and she's tall. And my dad, let's make him homozygous recessive, so he has 2 little a's and he's short. So to set up your Punnett square, you put 1 parent here on the top box. So I'm going to put mom up here, so I have a big A and a little a right there. And then I put dad on the side, and his was little a, little a. And then all I do is drag and drop. So if I look in this box right here, I drag the big A, I drag the little a, so I have big A, little a in this box. In this one, I drag him down, I drag him over, little a, little a. In this box, drag him down, drag him over, big A, little a. And then I drag him down, drag him over, little a, little a. So if this mom and dad had a child, this Punnett square shows me the possibilities of genotypes that they could have. So if I look at the Punnett square, right, I see that this one's heterozygous and this one's heterozygous. So 50% of my offspring will be tall because at least half of my boxes have 2 big letters. And then if I look at these two both have 2 little a's, so that means 50% of my offspring will be short. So if they have 10 children, 5 of them should be tall and 5 of them should be short. Or I can talk about their genotypes, because that's phenotype right there. I can say 50% of them are heterozygous, because look, I have 2 boxes with a big A and a little a. Or I can say 50% of them will be homorecessive, because I have 2 little a's in 2 boxes. All right, let's do another example. Let's do eye color for a trait. Okay, I have brown eyes, which is going to be dominant, and then blue eyes is recessive. So big A will give me brown eyes, 2 little a's will give me blue eyes. And let's say both of my parents, my mom and my dad, both have brown eyes, but they're heterozygous, so they both have a big and a little letter. So what I'm going to do is set up my Punnett square. So I drew my box. I put one parent on the top, so big A, little a. I put one parent on the side, big A, little a, and then I just dragged and dropped. So drag him down, drag him over, big A, big A. Drag this guy down, drag that guy over, big A, little a. Drag him down, drag him over, big A, little a. And then drag him down, drag him over, little a, little a. So this box shows me the offspring predicted genotypes. If I think about what they look like, my phenotype, I have 3 boxes that have at least 1 big letter in them. That means 75% of the offspring are going to have brown eyes. 75% of them will have brown eyes. And then if I look, this one has 2 little a's, so 25% of my offspring will have blue eyes, right? So if I had 100 kids, 75 of them will have brown eyes, but 25 of them will have blue eyes. Or I can look at genotypes. I can say 25% of them will be homodominant, because look, I have 1 box with 2 big letters. I can say 50% will be hetero, because I have 2 boxes with a big and a little letter. And 25% will be homozygous recessive, because I have 1 box of 2 little letters. And that's how I can use Punnett squares to predict offspring genetics. Now, one little thing, in addition to it, we can also look at ratios if you don't want to do percentages. Ratios, so instead of adding these boxes up to be 100, you just add them up to 4. So a genotypic ratio, I have to look at my genotypes. So I'll look at homodom to hetero to homorecessive. If I look at this box right here that we just did, right, this one right here, let's count how many boxes are homozygous dominant. How many boxes have 2 big letters? 1, so I have a 1 right there. How many boxes are hetero? That means they have a big and a little, so I have 1, 2, there's 2. And then how many are homorecessive? That's the 2 little a's, that's 1. So my genotypic ratio would be 1 to 2 to 1, which will add up to 4, because I have 4 boxes. Or if I look at this one up here, okay, I want to look at homodominant. Do I have any homodominant in this box right here? I don't. So I just leave, that blank, right? I don't put anything right there. And then I want to see how many are hetero. I have 2. And how many homorecessive? I have 2. So that genotypic ratio would be 2 to 2. And then phenotypic ratios, I do the same thing. I just look at dominance versus recessive. So if I look at this box right here, this very last one we did, how many show the dominant trait? I have 1, 2, 3. 3 boxes show the dominant trait. So that's 3, and then 1 box has the recessive. So my phenotypic ratio would be 3 to 1. If I look up at this box, how many show the dominant trait? 1, 2. How many show the recessive? 1, 2. So my phenotypic ratio for that first example would be 2 to 2. Okay, so that's pretty cut and dry as far as genetics goes. I just need you guys to remember that you get 2 copies of every gene. That's where diploid comes from. Genes code for traits. Trait will give me a physical characteristic. I can have different versions of a trait. That's where an allele comes in. I use letters to give me alleles, and I use capital letters to be dominant. I use little letters to be recessive. Since I get 2 copies of every gene, that's why I get my genotypes, which is a combination of 2 alleles. I have homozygous dominant, too big, heterozygous big and little, homozygous too little. But since I can't see your genotype, I can also look at your phenotype, which is the physical expression of your genes. And that's when I kind of start talking about like tall or short or brown or blue eyes. And then I use Punnett squares to predict offspring genetics. All right, I hope that helps.